Good morning. I think we'll get started now as people keep coming in. So this morning we have a really wonderful first session to wake you up. Peter Kharchenko is here from Boston, and he's talking about the extremely important subject of how we can put complex data sets together to use them.
Thank you, Sam. All right, so well, thank you, Fabian, for inviting me here, giving me an opportunity to present, and having all these nice venue, nice sort of tricks. I really liked your word cloud yesterday, and I'm glad that the integration was on the board there very prominently. It gives me an excuse to present our integration method, and it seems like these days everyone has their own. But we've been pretty happy with it. In fact, it sort of took over pretty much all the projects. We're using it throughout most of the projects in the lab. And so I'll try to describe the principles and some of the caveats the way I see it. I think I'll stand here because I saw Christoph almost fall off the stage yesterday, backing up. All right, so first of all, the motivation, why are we doing this, right? So I think the general motivation, at least within the single cell RNA realm, is that the methods have grown up, essentially. And instead of kind of single-shot experiments where you simply demonstrate an ability to do measurement at higher precision and analyze a single sample, now we have more complex designs. And the real-world designs, very clearly will give you, let's say, case control studies, right? And you need biological replicates, that do have variability. In the sort of clinical setting, we're talking about patient panels, right? Patient cohorts, and inter-individual variability is quite significant. If you compare mice and humans, humans are quite a bit different in terms of their even expression state. Quite often, in studying a particular disease or trying to answer a question, you actually want to assay multiple tissues, and some of them are more or less independent in terms of composition, but there are some parts that overlap. You want to perhaps track the state of immune cells throughout these tissues and so on. Finally, you can have treatments, for instance, that can really perturb the tissue, change both the composition and the state of the tissue, and move some of the cells to aside in terms of transcriptional state to make them look less similar. Yeah, so the example I'll start with is actually one of our studies. Here we're trying to study the impact of prostate origin metastasis on the bone marrow. So essentially, we're sampling at this point, dozens of patients at different stages and the various types of treatments. And there are different types of samples. So for instance, we can get these surgical samples, which are essentially solid pieces of matter with plenty of tumor cells in them. And so here I'm showing 7 patients and separate t-SNE plots, separate independent analysis for each one. We also want to sample the bone marrow around it. So this is a liquid aspirate of the bone marrow at the site of the metastasis. Same thing we could do for distant vertebrate where we don't expect metastasis to have an impact yet, at least not a local impact. So again, we get another panel. You want to have controls, obviously, right? So in this case, we're looking at benign surgeries, so they're hip replacement surgeries. And again, we're getting a set of panels from different individuals in this case. Blood can be very informative, and you can sample it serially. So we actually have a collection of blood time points for these individuals throughout treatment. And obviously, blood is going to look different, systematically different from bone marrow. And so the questions that we're trying to answer, we want to know, let's say, the compositional impact, so which populations go up or down. And we also want to know the actual impact on the transcriptional state of individual cell types, right? So essentially, to do both of these, and eventually we want to study the effect of treatment, but I think for that we're going to need much higher numbers in terms of individuals. But essentially, for all of this analysis, what you need is a sort of a joint way to track a particular cell type or cell state throughout your collection, if it exists there. So if you have a particular flavor of cytotoxic T cells that you're interested in, and they occur in a fraction of these samples, you'd like to be able to label them clearly. So it's kind of a joint clustering, essentially, problem. So now I've recolored all these samples to have a consistent clustering, essentially, and consistent coloring. And you can start, to see some of the patterns basically emerge in terms of sequences of states and so on. All right. So first, just wanted to kind of roll back and talk about problem formulation a little bit. I'll try to emphasize it. So I think the obvious formulation that we're most used to is batch correction. In this case, we typically talk about technical effects, such as, let's say, different sequencing run or perhaps different library prep from the same sample. But essentially, we assume that the samples generally will be pretty similar to each other, and whatever difference is there needs to be eliminated because it's technical in nature. The other sort of intermediate state has been explored by a couple papers last year from Raul Satija and John Marioni's groups, and they called it sample alignment. And so here, for the most part, either by doing these types of transformations, CCA, or doing this mutual nearest neighbor reciprocal best hit mapping, they're able to kind of align or establish mapping between reasonably similar, usually sets of samples in terms of composition, and then bring them back to sort of a single expression space. And that's been obviously very successful and widely used. So we're going to try to extend it a little bit to sort of joint analysis across very diverse collections. And when you do this, then I think the aims can change a little bit. So first of all, let's just take this simple patient panel, for example. So you have multiple individuals, and you have bone marrow measurements for them, and they vary, right? And depending on the state of individual, their genetics, and so on. So the notion of this common expression space becomes a little bit fuzzier. What does it actually mean? And then in most cases, what you're interested in actually is variation between, let's say, between different individuals, or between case control studies, and so on. So I think in our approach, we basically try to avoid this notion of common expression space and simply have sort of flexible mapping between data sets and between subsets of cells that could be used for follow-up analysis. So the basic idea is to kind of wire the data sets together into a joint graph that will actually include all of the cells. All right, so how do we do this? So the first step will look familiar. Essentially, we do kind of a pairwise alignment of two samples, and we do an error-prone mapping between them. So we tried a number of approaches. I think initially we focused on this GSVD and then this common principal component analysis as sort of good rotation strategies for aligning a pair of samples. It turns out it doesn't actually make that much difference at the end. In cell mapping, initially we started with nearest neighbor, and then when John's work came out, showing reciprocal best hits, so we switched to that, and it seems to be actually an improvement. And so you do this kind of a pairwise alignment, but essentially you expect to make mistakes there all the time. And so the main thing that we're trying to do then is to do many of these pairwise alignments between essentially all the samples and the data sets, right? So it's a pretty simple idea. And if, let's say, this blue population is indeed a recurrent population throughout the collection, it will tend to map to each other across all these pairwise collections, forming this kind of a clique in this graph, right? So we have this joint graph in the end that links all the cells, and subpopulations appear as these communities in the graph. And if you have more samples, here we have only 3, but let's say we have 12 samples. You have about 100 pairwise comparisons, and across these pairwise comparisons, you basically get a very strong statistical signal that a particular subpopulation is recurring. All right. Yeah, I just said that. So we also add these local edges to at a lower weight to kind of maintain integrity of some of the subpopulations that are perhaps not seen in other samples, and that's helpful for stability. So once you have this joint graph, you can use it for a variety of purposes, and I'll illustrate this. Obviously, you could visualize it, and just as a way of making embeddings. You could do these joint clusterings on it. You could propagate information between samples, and so on. So here's the first example. This is HCA data from the Broad. We have 8 bone marrow collections and 8 cord blood collections. And this is actually a visualization of the joint graph, right? So we simply use, in this case, large viz, an embedding algorithm that just projects the graph here. And with colors, I showed the joint clusters. So this is mixing, actually, of samples. So you see a strong bias here, but most of this bias is due to the bone marrow versus cord blood, actually, because there are distinct tissues, after all. And obviously, you could then look at each individual panel and color it by the same joint clustering if you want to focus on individual subpopulations. We did some benchmarking, and in this area, to be honest, there are no sort of consensus benchmarks at this point, so we're kind of looking forward to actually developing better ones. But the 2 properties we were looking for is stability and sensitivity, right? So in this case, we tested stability under various perturbations. For instance, we could take this data set as it is, and trust that when you have a full data set, any sort of integration method will do well. And then we start subsampling cells, and we see how well the clusters hold up. So that's, let's say, robustness. The other one that we're actually more interested in is heterogeneity. What if we start to increase heterogeneity between the samples? We take out some of the subpopulations from some of the samples, but leave them in others. And again, quantitatively, we do this by increasing probability of emitting a particular cluster, random cluster from a given sample. And we see how different methods hold up. Obviously, to this, you have a kind of a complement, how well bone marrow and cord blood, for instance, to different tissues, how well they're mixed between each other and within each other. So, it does reasonably well. This is just a quick thing to emphasize that actually most of the performance gains, they come from the fact that we're doing so many comparisons. We have this kind of a joint network representation. You could change to different sort of spaces. As I mentioned, PCA, for instance, combined PCA works pretty well, and it's much faster than CPCA. You could actually use gene space in a lot of cases and still works. Mutual nearest neighbor and nearest neighbor. So mutual nearest neighbor works better, but even with nearest neighbor, so this is basically same data reanalyzed, you still get reasonable results. One property that I really like is what we were kind of striving for, is that as you add more and more samples, let's say in this case they're pretty uniform with testing blood, right? So you add more and more blood samples from different individuals, and they vary obviously. But as you see more examples, you're able to recognize and resolve sort of more and more recurrent subpopulations. So the idea is that if as we get sort of larger and larger collections from different sources, we can start to integrate them and basically increase our statistical power to actually detect, distinguish finer recurrent subpopulations of states. All right, I'll show a few examples, and then I'll go to more exotic examples. So this is a simple one. So we're reanalyzing a data from head and neck tumor. And these are the individual samples that we're looking at. And some of them, what I want to point out, are sort of low complexity samples. So those tend to be problematic because they don't have much heterogeneity within them. It's hard to tell, whether, which subpopulation this may be. And this is the integration here. So again, this is the provided annotations, and this is how the samples end up mixing. And you can analyze the composition of each joint cluster that you get, and so on. So generally, we did the number of these, I think, about 10 different data sets from different studies, 10 different collections, right? And we tend to recapitulate the major subpopulations in the publications quite well. Okay, so another example here, also tumor-related breast cancer. And this is sort of a more advanced design, a more complex design where you have multiple tissues sampled from lymph node, from blood, and from normal, different individuals, uneven sampling. So it's kind of a complex collection. So what I wanted to emphasize is that when we show these types of joint clusters, actually, that a good way to represent them is quite often through a hierarchy. Because let's say T cells, they separate as a group, and then within T cells you have different subpopulations. So here I drew this hierarchy. And what I'm showing with the thickness of the line is the entropy. So this is how many samples essentially contribute to that particular branch. Right, so the thick branches are well mixed, and they have many samples and probably many tissues contributing. Whereas thin lines, they tend to be isolated from a specific sample, a specific individual. I'm also going to color them. So here, because there are a number of tissues here, I'm basically coloring tumor, blood, and lymph. And so, some of the edges, let's say here, they're pretty well balanced. So essentially, all tissues kind of contribute there. But the point is that if you go down, typically, in the hierarchy, so let's say if you zoom in and start looking at T-cell subpopulations, you'll at some point start hitting these branches that are tissue-specific. So let me give you an example. This particular node here, if we cut it here, we're essentially distinguishing CD8 and CD4 T cells, right? So very crude sort of resolution. If we cut it lower, we're going to distinguish all sorts of substates, but a lot of them will be tissue-specific. And this is, again, where we have to kind of grapple with what question is it that we're trying to answer here. Are we trying to make all sort of T cells, let's say all CD4 T cells, look the same when we're aligning? Or are we trying to basically keep some of the biological distinctions? Because we expect, essentially, let's say, T cells from one tissue to another to be systematically different, right? So if we ideally mix them in a single cluster, we're losing some biological truth there. But in other circumstances, they may be desired. So, I think, you can't answer this algorithmically, obviously, because it depends on the question being asked by the researcher. So we came up with sort of several strategies to allow control over this trade-off between resolution and breadth. So breadth is how many samples, let's say, or how many tissues you cover, or conditions, and resolution is how fine of a subpopulation you're able to actually jointly analyze throughout the collection. One way is to basically just look at this hierarchy and let the user go through it interactively and analyze at what level they actually want to interpret, let's say, T cells in this study. So for that, we just built a little interactive app where you could actually cut it at different levels and look at sort of the results and get some diagnostics on what's happening. So a more algorithmic way is to, if you know what you want, is to essentially force the alignment or force better mixing of either all samples or particular sets. So here we're using this classic, at this point, example, the collection of different pancreas islet data sets, and you heard some of this biology before yesterday. And if we just simply take it from these 3 different protocols, you see that, yes, different cell types are separated, but within each you kind of have separations, subpopulations that are essentially 10x, SMART-SIG2, or interop, right, so platform dependent. Because again, these are systematically different, and if you have multiple 10x samples next to multiple, let's say, SMART-SIG2 samples, the 10x samples for the same cell type will tend to be a bit closer to each other. It's normal. But in this case, we do expect this to be purely technical, and it's reasonable to say that we don't want this level of separation. So you should be able to control that. So we have a couple of strategies that play into this, for essentially forcing the alignment. One is to essentially increase the neighborhood size. So normally here for this, let's say these 2 samples, when we're comparing them, when we're looking for potential matches to these blue cells, we go here on the other sample and we look at the radius, let's say the inner circle in this case. And then based on that radius, that neighborhood, we determine reciprocal best hits. So we can increase this radius, essentially. We don't want to increase the complexity of the graph. In fact, we want to kind of relax it a little bit. So we can take a larger radius and then sample, almost randomly sample edges from that to get these sort of looser best reciprocal hits. So essentially, we're kind of defocusing the mapping between 2 individual samples. So when you do this, so here I'm increasing the strength. When you do this, slowly, at first they're still separated, and here they're also separated, and then they start to mix, and then you could show this quantitatively. Of course, if you take it to the extreme, that's what you're going to get. And to me, this is kind of an illustration of that trade-off, right? You're losing all the resolution, but everything is perfectly mixed. So the other thing that happens here, and I'm not showing it separately, is edge weight rebalancing. So this is a weighted graph, obviously, and we can do a lot of things on this. And particularly, you could look at this weighted graph and with a notion that you have a confounding variable, let's say platform in this case. And you can actually optimize the weights in a way that makes sure that overall in the graph, they don't depend on that confounding variable. So this way, you essentially erase that or try to control for that platform difference explicitly. And that helps mixing as well. Okay, so other uses of the joint graph, just think it's a useful representation, but there are practical things that we'd like to do as well. So you can essentially use this graph as a sort of scaffold to propagate information between samples that you have or between cells, either discrete or continuous values. We do this through basically by modeling a diffusion process. For discrete labels, I'll show you, for instance, just annotations. That's an easy example. For continuous ones, it will be actually, for instance, these common expression coordinates or some other values. So let's say for annotations, this is the bone marrow HCA data set again. And so we just took the bone marrow, 8 samples, and now we're going to pretend that we know annotations only for this one. So we forgot annotations for the rest, and we tried to recover them. And this is basically labeling of correctly recovered annotations, incorrectly recovered, because we knew the answer. And you see that there are a number of red cells, but they actually appear at very specific places. They appear at the boundaries of clusters, for the most part. And, or some of these sort of presumably doublets that are uncertain to begin with. So, you know, that's expected. At the boundary of a cluster, you may not be able to classify a cell very well. So we can actually quantify this, because when we're doing diffusion, we can keep track of uncertainty of the label, right? So you have a combination of labels that arrived at a particular node. And so here, just with a heat map, I'm showing uncertainty. And so what's easy to see is that basically the places where we made a mistake, we actually thought that we're likely to make a mistake because we had high uncertainty there. So that's actually a useful feature. And nowadays, when we're interpreting clinical panels, this is essentially how we do annotation. We align it with something, public data set or something else that's annotated, and quickly transfer the labels, and then consider whether actual annotation was correct or not, instead of looking at marker genes. Yeah. So as I mentioned, we don't actually find much use for these common expression coordinates, to be honest. But you could do this. And so here is a sort of illustration. This is basically taking again, these bone marrow data sets and simply not doing any corrections, right? And when you do this, essentially everything kind of clusters by sample. That's the traditional thing. And so, but if you do these, the same analysis, but in these common expression coordinates, things are much better mixed, which is what you would expect. But I want to caution again, against sort of using these types of things, especially for something like, let's say, differential expression analysis, because essentially, first of all, if you're comparing between individuals, the common coordinates explicitly erase that difference, obviously, so you don't want to do that. But if you're comparing between conditions, that would essentially over-smooth the data, or smooth it to some extent, reducing variability on each side, right? So let's say if you had 5 individuals on normals and 5 disease, you actually want to take into account the variation within disease, within normals, but common expression coordinates would reduce that variation, inflating your p-values. Okay, so more exotic examples, atlas collections. So this is tabula Murris illustration, so they're basically sampling different parts of the mouse. And it was amazing data set, very quickly put together. And they used 2 platforms. And obviously, these individual samples, they can look at very different parts of the organism, and the composition is very different. But there are some cells, let's say immune cells, that would occur in many tissues, although their state would be quite different. So can we use the same approach to basically wire it together? So here we started with just tabula Murris, and it's 48 data sets, about 100,000 cells, and this is just visualization of the joint graph. This is actually the platform factor on that joint graph. So SmartSeq2 and 10x were the 2 platforms used. As you can see, there's some places where they're mixed, but in most of the places they're not. And that's expected too, because essentially 2 platforms were used to sample different tissues. So there's not actually much overlap there. And then this is just projection of their annotation on the same joint graph, just to show you where things are. But we'll make it a bit more complicated first. So this was the initial atlas, but in parallel, actually, with that, there was another atlas from Geji Gur's lab that was published using microwell technique. Much lower coverage, sort of higher noise, but more cells and more data sets. So in this case, we basically have 127 data sets, about 400,000 cells, and this is just the projection of joint clustering. And it gets rather complicated, so you kind of have to look at it as sort of abstract art at some point. But we can clean it up quite a bit. So one approach to clean it up is what we call decoy cells. And so the idea is that if you're comparing 2 samples that are quite different in composition, so in this case, this blue subpopulation is missing from this sample. So most of the time, it will be OK, let's say, if you're using reciprocal best hits, because it won't be able to form them very well. But once in a while, it will form these edges by error, right? So that's why we expect an error-prone alignment. Now, if you're doing a lot of pairwise comparisons, statistically, that doesn't matter. You can tolerate these, this type of sort of edge noise. But still, we'd like to clean it up. So what we can do is kind of an iterative approach, where in the first analysis, we can confidently deduce that this sample was missing a blue population. And then we'll redo the alignments between this pair of samples. And when we're doing this, we know that the sample is missing blue population, so we're going to add it from another sample. And these are decoy cells, so essentially they're designed to sort of attract mappings of other blue cells in the other sample, and we know that all these edges, they don't have to be in the graph, so we can remove them and reconstruct the graph. And that cleans up some of those spurious edges and basically gives you a cleaner layout. So this is, again, this 127 data set collection, just with annotations, I think, from Tabula Muris. And then the graph structure looks a lot more sane from biological standpoint. So here, for instance, hematopoiesis, I think, quite nicely laid out with neutrophils and T cells here, B cells, branch, and erythrocyte, and so on. And of course, there are isolated populations still that you would expect. Okay, so the primary motivation really for our study has been these types of, let's say, clinically oriented collections where we're working with the same modality. And as was mentioned yesterday, I don't think you should plan on a design that has multiple platforms and so on. But multiple modalities is something you would probably want to measure just because it gives you complementary information. So you could apply, let's say, the same algorithm to a different data type. So here we took the ATAC-seq atlas from Jay Shandur's lab. So this is basically 17 ATAC-seq, single-cell ATAC-seq data sets covering 13 tissues, and simply ran the approach on that data. So again, it combines, it recognizes the subpopulations that have been annotated by that paper. This is mixing of the data sets. These are the 17 data sets. And some places are well mixed. Usually the immune system is well mixed, but others are not, because again, let's say hepatocytes were sampled only in one sample, so you would expect that. Okay, so this is simply showing that if you apply it to, let's say, epigenetic data, it seems to work reasonably well. But what we actually want to try, probably more interesting, is integration, let's say, of RNA and ATAC-seq data. So here again, we're using for ATAC-seq that same part of that data set from Jay Shandur. And then for RNA-seq, we have these mouse atlases, right? It's all in mouse. So this is an example from lung. Again, we have the RNA data with the RNA annotation from these two atlases. So I'm just showing both of them. That's why it's a bit messy. This is the annotation from J for ATAC-seq cells. So I'm just showing the cells separately, but it's part of the same graph, right? The same embedding. And this is sort of platform or data set annotation. You have a couple of ATAC-seq data sets, and you have all these RNA data sets, let's say, with micro-L, SMART-seq2, and 10X. So we have 3 RNA-seq platforms and ATAC-seq. You could see that, again, there's a similar effect. So for instance, let's say these T cells, right? You see kind of a separation, even though it laid them out pretty well, but ATAC-seq samples a little bit to the right, and RNA samples also kind of layer in a little bit as well, because again, there are consistent differences between them. And nevertheless, it's able to recognize, let's say, NK cells versus cytotoxic T cells. It kind of stretches out this cluster. That's what we'd like to see. All right. Now, the key thing here is how do we actually do this? And people don't discuss it, I think, when they try to integrate this. Now, we do this at a gene level. You need some sort of an initial clue to link the, let's say, your gene expression space with your accessibility space. And we know enough biology here to quantify this reasonably well. So a simple sort of gene level accessibility score could be simply, let's say, accessibility at a promoter site, right? So that would be a very simple one. In this case, we started with a Cicero implementation, which is sort of much fancier way of scoring different enhancers associated with the genes, and gives you probably more informative scores. But you could simply take, let's say, whole gene accessibility across the peaks, and it works just as well. But you could degrade it quickly. So let's say if you take just around TSS, around promoter side, the accessibility, and start narrowing down the promoter window. At 10 kb, it still kind of works, but let's say at 4 kb around promoter, you don't have enough signal to start establishing that kind of a mapping. And yes, I'm done. Last slide, I think. So I just wanted to, I think, caution that you can have a beautiful kind of integration algorithm, but its performance for its ability to link across modalities will depend on that initial sort of clue of how these spaces are potentially aligned. All right, so summary, basically, it's a pretty straightforward approach to wire together all the cells in a collection for the purposes of joint analysis, whether you're doing clustering, expression differences, and so on. and I try to illustrate that we think most of the sort of statistical power and stability comes from these multiple comparisons that you essentially reflect one sample on another many times across these pairs, and that allows you to get a stable picture out of this. You know, we can apply it to sort of all kinds of challenging cases, different tissues, sort of atlas-like collections. And this other property we really like, that the power to recognize subtle subpopulations seems to grow with the collection size. And finally, wanted to acknowledge the people who worked on this. Again, this tool kind of arose as part of collaborative projects like this bone marrow project here. So Nick Barkas worked on this quite a bit. Victor Peterhoff, who is here, has been instrumental in the second part, and I encourage you to talk to him, and as well as some of the folks mentioned here. Thank you very much.
Thank you. That was a wonderful talk. We have maybe a minute for questions if there. Fabian.
Can I get that? That was really obviously it was very nice, tour de force, and we enjoyed that. Just first, can I understand this one? I mean, you showed this once before, but you have this reciprocal hits, right? I didn't understand how you take care of sort of populations that only pop up in one thing. You mentioned those decoy cells only later, but even in those early data sets, it could happen, right? There's this one population missing, I think, in those pancreas islet data sets.
Yeah. Well, there are extreme cases. In essence, this method and most of the others, they kind of make an assumption that the extent of expression difference can be arbitrary. And what actually matters is sort of a structure of the subpopulations. So the reciprocal best hits, the reason they clean things up is because if you have heterogeneity within one population and another, the probability of you actually getting the matching pair is becoming lower. However, if you have this, like I showed, in one of the samples, for instance, you have this kind of a round cluster. So essentially, you have a single cell type. Then you don't have enough heterogeneity. Then you have to rely on similarity or heterogeneity in the other sample to get the correct mapping. So the reason we're able to map those round cases, which are the most challenging one, is because we're comparing it with other samples that have considerable heterogeneity. And in that context, we can kind of guess, which sample, which cluster it should belong to. And again, if you do many of these comparisons, then that becomes a very strong feature in the graph, and you can map it correctly. Now, if it's completely separate, essentially it will have spurious edges between the samples, but there will be very few and non-specific in terms of any attracting the cluster to any subpopulation. And that's why we keep these within dataset edges. They're really weak, but they allow that subpopulation to essentially just stay together as outside of others. So that's how we get robustness for these exception cases.
Any other questions? We can take another minute. There's one over there. Oh, Rob.
Thanks, really nice talk. I was wondering, I didn't quite understand how you use the locality in your individual samples, like your edges, as you mentioned. When you relax that constraint, you showed kind of the really big blob. Is that some kind of uncertainty you allow on the mutual nearest neighbors, that you don't have to map to exactly the same cell, but rather in the same locality, or how exactly is that work?
Yeah, so this has to do with this kind of trade-off of what do we mean by alignment? What do we actually want to do? And so by default, you set some kind of a neighborhood that allows to establish connectivity within the collection, right? But not larger than that, because you don't want to lose resolution. And what often happens is that you find these smaller subpopulations that are, actually distinct. Let's say, disease versus control within each cell type, or different tissues within each cell type. And when you don't want this, what essentially you need to do in order to kind of establish connectivity is look further out, right? Instead of that exact sort of best match, you need to look further out. And so this control essentially allows you to do that. It kind of defocuses that mapping. So you look at a larger neighborhood and then sample from that, again, the reciprocal best hits. So you still maintain this kind of structural robustness because you're not mapping erroneously to a different population. You're just mapping to a broader region, around that population that you think is best hit. And that allows you to mix these types of effects. Now, edge rebalancing is more targeted. You could say that I have factors I want to control for, and on top of that graph, it obviously affects both the clustering and other properties.
Thank you.
Thanks.